Okay, hi everyone. This lesson is going to start looking at more than one trait. So, thus far, we've only talked about one trait at a time, which you might remember was called a monohybrid. So, differing by just the one trait. And they were only controlled by one single gene, or that's what we assumed through doing our Punnett squares and our problems. Now, what we're looking at is Mendel took it to the next level. He extended that further and he started to look at two traits at a time rather than just one trait at a time. So I'm going to go back to what we talked about in Mendelian genetics, rather, which is some different traits he looked at in pea plants. And again, just reminding you, part of the reason he uses pea plants is they're really quick to reproduce. They produce many offspring. So I was talking about before, the more offspring you can take a look at, the closer to predicted results you'll actually get. He can also really easily see the traits that we're talking about. So again, he did this long before we knew about meiosis, long before discovery of DNA as the hereditary material. But we're going to try to make those connections now back to meiosis. And now we're looking at two traits at a time. So we call that a dihybrid. So I've titled this as multi-trait inheritance, meaning more than one trait at a time is being traced. If you go on to take genetics after high school, you will find that it gets much more complicated. So you can get into three traits, you can get into even more traits. And there's a mathematical way that we can make predictions, which I'm going to show you the basics of here. Also relates to a lab you're going to do on gizmos again. So this lesson will be really important for setting you up for that lab. Okay, so I'm going to show you what I mean by the dihybrid. And I'm going to continue using these alleles for most of the lesson. So we'll just use our let statements right here. So we're going to still be using complete dominance, which is that one of the alleles is dominant, the other one recessive. We're going to assume it's only controlled by those alleles that we're mentioning here. So I'm going to use small case r for wrinkled seeds. That was the recessive phenotype, if you remember from Mendelian. And capital R is round seeds. Small g is for green seeds, large G is for yellow seeds. So this goes back to alleles that we listed like long ago in Mendelian genetics. We're using the same dominant recessive, but each question would tell you individually, which I'll show you in the examples. Now, really important in this whole concept is this note that I've put at the bottom. We are assuming that these alleles are on different chromosomes and therefore are not related to one another. So what I mean by that is just because you are a wrinkled seed does not mean that you must also be a green seed. Okay, these are totally independent of each other, which I'll explain in just a few minutes. Just to show you, the genotype looks much different now. When we're dealing with dihybrids, we're going to have both traits represented in the genotype. So where before we would only have, let's say, the shape, the wrinkled or round, now we have actually both traits. So our genotypes, when we're writing dihybrid, we should have 4 alleles shown, right? 2 alleles for each trait. So 2 for the shape, 2 for the color. Now I've written on the side, this particular genotype represents heterozygous parent for 2 traits, which is they have 2 sets of chromosomes still, one from each parent. What I'm going to show you in this part, Mendel's law of independent assortment, it's going to go back to something we discussed in meiosis, which is independent assortment, also called random assortment. So what we see happen here is that the heterozygous individual is going to produce 4 unique gametes, okay, 4 different gamete combinations. I'm going to show you how that works in just a second. The alleles for small r and capital R and G and capital G separate independently during gamete formation. So again, I'm talking about anaphase here. Whichever direction that they go in is totally random, just based on how they've lined up in metaphase. Each allele is independent of each other and on different chromosomes. So that was my note that I mentioned before. We must assume that, otherwise we cannot use this type of method that I'm about to show you. So as mentioned, it means that the inheritance of a particular seed shape has no influence on a particular seed color. Again, we're oversimplifying things. This does not work for human genetics as well as it works for other things. We do have one really good example in human genetics, which is blood typing, which I'll show you in a few minutes. But what I want to show is like what I've done in this diagram. So I've done 2 green chromosomes, which are homologous to each other, and 2 blue chromosomes, which are homologous to each other. The reason I've shown this is to show where I'm getting this genotype from. So I have the allele small r and capital R. Those are for the shape. Those are on, again, the homologous chromosomes to one another. And then here I have small g and large g, again, homologous chromosomes to each other. So what I'm showing on the side here is one potential way that during anaphase 2 that these chromatids can be pulled apart. So the R's on the green chromosomes, they're going to assort from each other. They're going to separate from each other. Again, assuming no non-disjunction or any of that that kind of stuff. And same thing with the G's. They're going to assort independently during anaphase. So I'm just showing anaphase 2 here, but of course in anaphase 1 there is a randomness to how they align and how they're going to separate as well. Just for simplification, I'm not showing all of those steps. So the gamete that I'm describing here that would form, I would have capital GR on the left side, and on this side, on the right side, I would have small g and capital R. So again, I'm going alphabetically here. This is why I said it's sort of important to keep things alphabetical. Now, what I'm showing in gamete 3 and 4, these are the other combinations that might be possible. So I'm not showing that in this picture, but just thinking about it, it's very possible that these 2 blue chromosomes switch places. And so I would end up with the capitals in one side and the lowercase in the other side. So in total, for 2 alleles that we're looking at, R and G, there are 4 different combinations. And I'm going to show you like a trick afterward where you can just really easily figure out what those combinations are without actually drawing a picture of it or drawing a diagram of it. So what I'm going to be showing now are 2 different methods, and the 2 methods are essentially using probabilities in order to figure out the outcomes of a dihybrid cross. So when I'm now looking at 2 traits at a time as opposed to 1 trait at a time. So our 1 trait at a time would be our normal Punnett squares that we've been doing from Mendel. These are now when I'm looking at 2 things at a time, so it becomes a little bit more complicated. So method 1 is my preferred method, and I'll explain why as we go through it. We're going to be using probability and something called the product law. Okay, product law is something that you will learn about in data management if you decide to take data management in grade 12. But it's a really important tool also for biologists because we're always looking at what are probabilities of things happening. So biology is a science where things are not exact, okay? We're dealing with only probable outcomes. We cannot necessarily say things for sure most of the time. So product law states that the probability of 2 distinct or independent random events, both occurring, can be found by multiplying their individual probabilities. So what I'm talking about here is 2 different outcomes that are possible. Event 1 or outcome 1 is I look at the seed shape, and event or outcome 2 is that I look at the seed color. So here I'm predicting outcomes of a cross between 2 parents who are heterozygous for both the seed shape and the seed color. I'm not going to write the let statements again. It's the same ones at the top of the page. So what I'm doing here is I'm showing the 2 events separately. I'm assuming that seed shape and seed color have nothing to do with one another. Okay, they assort independently. They're not depending upon each other in any way. So what I'm doing here essentially is I'm separating it into 2 separate normal Punnett squares. So I've underlined here the R's and the R's. So I'm essentially going to take those 2 genotypes and I'm going to cross them down here, which is in the blue Punnett square. So I end up with a 3-to-1 ratio of round to wrinkled, or I can write them as fractions. For this particular method, I will say I find it easiest to put them as fractions. Some people like to put them as decimals. That's also fine. I would not choose to put them as a ratio here, and I'll explain why on the side, why I've chosen fractions as opposed to something else. But here, fractions or decimals is the way to go, okay? The second event is looking at seed color, so crossing the G's together. So parent 1 has capital G, small g, parent 2 has capital G, small g. When I cross those, I again get 1/4 of green and 3/4 of yellow. So what I'm doing in this method, and that's the phenotypes on the right side, excuse me. What I'm doing is finding the probability of 2 events both happening. Okay, so again, event one is seed shape. If I'm looking at wrinkled and green, okay, the wrinkled has a 1/4 probability of happening. So I write that over here as 1/4. The product law says I can multiply these probabilities together to find out the overall probability of both things happening. So then I look in my second event, which is seed color, and I see it's a 1/4 of green. So I do 1/4 times 1/4. Going way back to like math in elementary school, when I multiply fractions, I just multiply numerators, multiply denominators. I end up with 1 out of 16. So that's a 1 out of 16 chance that the phenotype of the offspring is both wrinkled and it's green. Okay. Second scenario, which is wrinkled and yellow. What I've chosen to do here is take one recessive, one dominant. So wrinkled being recessive, yellow being dominant. Again, I take 1/4 because that's the probability of wrinkled, and I multiply that by 3/4 probability of yellow. And I end up with 3 out of 16. Again, fractions, multiply numerators, multiply denominators, end up with the answer. So I do that for all 4 different combinations that are possible, okay? And I end up at the end here with probabilities of each phenotype. Now, for this in particular, it's not useful to me to figure out the genotypes themselves, so I won't ask you to do that. It's really not useful for the offspring. What's more useful is actually this part, which is probabilities of each possible phenotype. So that is method one. That is the product law using essentially simple Punnett squares. The second method, which as I said is not my preferred method, but some people like it, the second method uses what's called an extended or an expanded Punnett square. So I'm going to show you that one as well. Again, I don't mind which one you use. In the lab, you're going to practice doing both just for sake of getting the feel for what both of them involve and kind of choosing which method that you like better. So method 2 down here, the expanded Punnett square, we're using the exact same parents as example number 1 or method number 1. So what I'm showing here is the cross. So I have the 2 dihybrids, both of them in this case were heterozygous for both traits. And what I've done here is trying to figure out the possible gamete combinations. So you might remember from grade 10 math, we have this little like math trick called FOIL, which is to essentially expand something. So what I'm doing here is I'm essentially multiplying, but I'm putting together the first 2 alleles, okay, which is in this case capital G and capital R. That represents my very first gamete possibility. The O in FOIL is for outside terms, so capital G and small r in this case. So I write that here. Inside or I is small g and capital R. I write that here and so forth. So what this represents is one allele from each of the genes from each parent. So again, if you go back to page number 1, I talked about the reason why this is that during anaphase 2, they will always split up independently of each other. So we essentially treat them as totally separate entities. Okay, they don't rely upon one another. Now, what do I use these gametes for? These gametes are the same for both parents, okay, because both parents in this case happen to have the same genotypes. That's not always the case. But now I'm going to make an extended Punnett square. I'll just mention quickly right here, this is extremely important for the lab. So I'm going to ask you for the lab to refer back to this lesson because it's almost identical to what you're doing in the lab. So this will be really, really helpful to you. What I'm doing is writing out those same genotypes for parent 1 across the top and parent 2 down the side. And I essentially make my whole Punnett square using the same patterns that we've done before. I'll just reinforce what I've said previously. I prefer to keep them alphabetically because they're easier to actually see what's going on alphabetically, and within those alphabetical groupings, also keeping capitals before lowercase. So what I'm essentially doing in this chart is I'm putting together all the possibilities that the gametes can make. And you'll notice it's 4 by 4. There are 16 outcomes that are possible. So when I said before, I found the other one way simpler. The reason is because this one has a lot more room for error. There's a lot more steps involved. So I write out all of these genotypes, which if you have questions about, I'm happy to go over. But essentially what we're doing is taking the first letters, which in this case is 2 capital G's, writing them in, then taking the 2 small r's, writing them in, and then repeating this 16 times for the whole table. So what I'm doing on the side here, on the right side, is then taking apart what are the possible phenotypes? Okay, so again, I've just chosen it in whatever order. So here I've chosen to do yellow and round. Well, what those genotypes look like is they're both dominant. So I have a capital G and a capital R that I've written in right here. And I'm essentially going through the table and I'm counting how many possibilities are there. So I've crossed those out in green, as you see here. Any that has a capital G and a capital R, I cross them out. And if I count them all, I see that it's 9 out of 16 in the Punnett square. Okay. Second possibility is green, which is recessive, and round. So I look through and I look for all the ones that are small g's and also have a round shape. They're in this bottom right corner. So I've crossed them out in black to show that those are 3 possibilities. So 3 out of 16. Yellow, I do the same thing, 3 out of 16. You'll see those crossed in blue. And then the last one is 1 out of 16. I've crossed that one in black as well. That's this bottom corner. So again, what I want to notice is that using method 1 or using method 2, the outcomes are the exact same, okay? What I find is this 9 to 3 to 3 to 1 ratio. And this will happen every single time that I have a dihybrid cross with 2 heterozygous parents. So just like we had the 3 to 1 ratio with 2 heterozygous parents in one trait at a time, monohybrid traits, in dihybrid, this is a very, very common ratio that we'll find, okay? This down here is just a little additional information, and I want to go back to what I was saying previously, which is we are making a lot of assumptions by doing these Punnett squares. We basically, for Mendel, because he used a really simple model, he used a really simple pea plant. He is looking here at discontinuous variation. Okay, he has 2 characteristics that have nothing to do with one another. Okay, they assort totally independently, and they have 2 distinct chromosomes. As I mentioned before, human genetics is much more complex. We often cannot do that type of prediction. So our predictions using Punnett squares, we're using discontinuous variation. Continuous variation, I'm simply defining it for you here. We cannot use the Punnett square to predict this, so we will not see this in any type of question or any type of genetics problem that we do. Continuous variation is looking at things that have more than one allele that's responsible for it. So I've used the examples here of skin color, height. Okay, there's lots of other things as well. Even our hair and eye color will do that. These are things where the phenotype is not just one outcome or the other. There is often blending that happens. So I used the example before of height, okay? So whatever my 2 parents' height is, I am not right in the middle of that, okay? So continuous variation means that there is lots of phenotypes that are possible. I imagine it on sort of like a number line. So if you have sort of 2 extremes of a trait, and then there's all these different possibilities that come between that. That is continuous variation. So the reason I am mentioning that here is because we really cannot use Punnett squares for this. And in reality, more of the human traits relies on continuous as opposed to discontinuous. Okay, so the other part to this is called additive alleles. So sometimes you have alleles that actually work together to give some types of phenotype. Again, not 50-50 necessarily. An example of additive alleles is you might notice that people with red hair often have freckles and a certain type of very fair skin color. So alleles for all of these things are contributing with each other to form a very typical phenotype that we see. As I said before, with continuous variation, it's not possible at all to use a Punnett square. The only ones we can use a Punnett square are for discontinuous variation, okay? So what I've done here is just a couple of problem-solving ones. Really, it's for you guys to work through the problems on your own. But what I've started to do in here is actually put in some problems that is not just 2 heterozygous parents, because like I said, that's always a very, very predictable outcome. Okay, here I have parents that are no longer heterozygous. They have other types of phenotypes. So I mentioned here is the term of true breeding, which we actually saw in lesson 1. So sometimes called pure breeding or true breeding, those are the same thing. It means that they're homozygous for those traits. So again, I've done my let statements here based on just the description. What I've underlined in green is representing parent 1, which I've written here, and in red, parent 2, which I've written here. So what I'm talking about doing here is, first of all, taking what the gametes are. So I have done this in a modified way, and I'm going to explain why I did that. So here, with this AADD, all capitals, right, all homozygous for dominant alleles, there is only one allele possibility, right? If I do my FOIL method that I showed before, I will end up with 4 alleles that are all capital A, capital D, okay? So instead of writing that 4 times in a table, I can just simplify it and write it once in a table, okay? So this parent will have 4 identical alleles, 100% the same. The second parent is homozygous recessive for both traits. Same thing, if I think about what allele possibilities there are, it's going to be 4 that are identical, all of them small a, small d. So to simplify things, I can just write it as small a, small d. So when I do my Punnett square, instead of drawing all 4 by 4 and then just finding the same information, which is 100%, all 16 of them would be heterozygous, I can just simplify it by writing this. So you decide if you want to use that. I find it much simpler, and it shows that you actually understand that they would be all identical. What I've done on the F2 generation is just take 2 of this blue parent and cross it with itself. So I am showing method 2 over here. Again, totally up to you. You can use, you can use method 2, the expanded Punnett square. I just find the product law is much, much simpler, much less room for error. And just to sort of reiterate again, I end up with that same 9:3:1 ratio because in F2, I'm using 2 heterozygous parents. So going on, this example here, it's the exact same pattern. Again, I just have different gametes that I take from each parent. And then when I cross them, I'm doing the same idea with the product law. If you wish to use the expanded Punnett square, that also works. Okay, you can do the same method as above. Just the same thing should be true. Your whole answer actually is going to look pretty much identical to this. The difference being that you're going to have whatever allele names that you've made it. So it won't be G and R, it'll be A and D or whatever names that you've given it. So again, you choose your own method. In the end, you should get the exact same outcome. So that's totally your call. I just find product law is much, much easier to use. So I was mentioning before, oftentimes in humans, it's way more complex. But we do have one really good example of dihybrid here, which is using both the blood types, which is the ABO blood group that we talked about with codominance, as well as using the RH factor, which is like the minus and positive. So reading this information, it's telling me that the ABO blood group and the rhesus factor are on separate chromosomes. What that means is that they do not rely on one another. They do not travel with each other in meiosis. They assort independently, okay? So what I've asked to do here is, or what I've said here rather, in underlined in blue, is that the RH plus allele is dominant to the RH negative allele. So I've written just a let statement for that. Small r represents negative rhesus factor, and capital R represents positive rhesus factor. Just as a bit of an aside, the rhesus factor is just another antigen that's on the surface of our blood cells. So it's just in addition to the ABO blood groups. There's lots of other stuff there as well, but not as interesting to talk about. And then I start to pull apart information about the parents. So I have a woman with type O, Rh negative. So I've written her in red here, as you see underlined. So small I, small r, small r. Right? She's got type O blood, which is small I, RR, and that's what I've written here. And same with the father, homozygous type A and heterozygous for Rh positive. So I'm taking apart these gametes now. So the 2 small i's, because she's homozygous for both traits, there's only one possibility of the gamete, which is 2 recessive alleles. The father has 2 possibilities now because he only has I capital A, so that's fine, but he could have it in his gamete with the capital R or with the small case r. So he has 2 possibilities I've listed here. And then I simply make my Punnett square. So just as before, there's no need for me to write it 16 times because in reality, I'm going to get this exact same outcome. So I'm making it much simpler for myself rather than writing this unnecessarily. So what I get at the end of the day, which I've written in blue, this is the outcomes for the offspring. I can see that all offspring will have type A, okay? But the rhesus factor, I see 50% are going to have Rh positive, right? So A positive versus 50% have Rh negative, which is type A negative. So I list those two phenotypes off to the side, okay? Again, here we don't so much care about the genotype part, but we do very much care about the phenotypes. Now, the reason I put this little diagram is just for interest's sake. Why do we care about this? So in this particular scenario, the mother is Rh negative. There's a 50% chance that the fetus is Rh positive, okay? Now, why I've put this in, it's more just for interest's sake. It's not so much for the genetics, like problems and such, is that unfortunately with these pregnancies, excuse me, the mother will start to make antibodies against the fetus. And so the mother's body actually might start um, fighting off the developing fetus. So I mentioned here that because they're opposite to each other, the mother recognizes it as non-self. And so in her first pregnancy, usually nothing much happens with that. Her body is just sort of like identifying that it's something unknown to it. But by the time the second pregnancy rolls around, if she also has an Rh positive fetus, her body has had lots of time to make antibodies. And so we were talking before about sort of fertility and those types of things that people might seek help for. Well, this is one where definitely the mother would have to be monitored. And essentially, what the doctor will end up doing in most cases is actually giving her an antibody. So essentially, a drug which is meant to block her antibodies from interacting with the fetus. So again, this is kind of like where the real world application of this comes up. It's sort of like, well, why do we even have this, right? Why do we even care about this information? Well, it's really important because this mother could be facing a miscarriage if she's unaware of that problem. So any kind of information that we have about mother and father, we can actually use that to our advantage to help actually make it a successful pregnancy. So there's a few more examples for you guys to do, which is listed on the schedule. As I said, this lesson is super important for the lab that you're going to be doing on gizmos. So please open it often and just check through what the different strategies are. It'll be really helpful for you for the lab.